Thanks so much, Jean-Baptiste and all of the organizers for the invitation to be here today. I think I was actually at one of the Mifobio workshops in Biarritz maybe 7 years ago now, right before I moved and started my own independent lab. And so it's fantastic to be back, fantastic to see everyone again, and to tell you a little bit about what we've done since moving to University of North Carolina. So I'm Wesley LeGant. I'm an assistant professor. I actually have a joint position in biomedical engineering, which is a school in the College of Engineering, and in pharmacology, which is a basic science department in the School of Medicine. And my lab right now is split between multiple different avenues of research. So about 30% of the lab is spent building and designing new microscopes. So from the ground up, new optical principles, Zmax simulations, light propagation, and assembling these into useful instruments, particularly light sheet microscopes. And then I disclose I am an inventor on patents related to the use and application of lattice light sheet microscopy. So I'll get that out of the way early. And then probably about 10 to 15% of the lab is focused on using these instruments in new ways, trying to increase the accessibility of the microscopes, enable new types of experiments with them. And then the remaining 60% of the lab is actually biologists through the School of Medicine using these instruments for various applications in cell biology. One application, which I won't talk about today, but sort of a carryover from my work during graduate school looking at cell mechanics and traction force microscopy, is trying to understand how cells move through complex three-dimensional environments that we can control and generate in the lab, and generating biophysical models for how cells navigate through these complex three-dimensional settings. And then a large part of the lab now is focusing on a, say, smaller scale question, which is understanding the relationship between nuclear organization and gene expression, and looking at how individual molecules like transcription factors or methyltransferases, acetyltransferases, navigate through this complex three-dimensional environment inside the nucleus of individual cells. And I'll give maybe 3 little vignettes today, talking briefly about the new technologies we're working on, a recent project using AI to teach them what to image, and I'll focus the majority of my talk, because this is a nanoscale imaging presentation, on our work using single molecule imaging. So I probably don't need to belabor this point. I'll go quickly through this, but one of the motivations for light sheet microscopy is that in contrast to confocal or wide field instruments that illuminate the entire three-dimensional volume of the specimen, every single time you take a two-dimensional image, light sheet microscopes can offer tremendous advantages in terms of photobleaching and phototoxicity by only confining the illumination light to a thin plane within the specimen that's coincident to the focus of, for example, a high numerical aperture detection objective. From this, then we can scan either the light sheet and the objective or the sample through this cross-sectional volume, capture a single plane of fluorescence at a time, and then rapidly assemble three-dimensional images of our volume of interest. So one of the areas that I had focused on in a postdoc and continue to work on in my own lab today is overcoming this challenge of a trade-off between the length of the light sheet that you can generate and the thickness of the light sheet. It's determined by the Rayleigh range. So this is an example of a conventional Gaussian beam. It's about 1 micron long, or sorry, 1 micron thick, which is what you would want to pair with the depth of field of a high numerical aperture detection objective. And the challenge with this is that that Gaussian beam will be about 10 microns or 8 microns long, full width half max. So that defines a very narrow stripe of a field of view that you can use for imaging. So when I was in Eric Betzig's lab during the postdoc, we started exploring alternative methods of generating other types of beams used for light sheet microscopy that could overcome or decouple this trade-off between propagation length and beam thickness. One avenue is a Bessel beam, which was published previously. This is actually challenging because it's this radially symmetric pencil, and effectively it's a line scan technique. And so the duty cycle is very, very low, photobleaching rates are higher, and there's all of this off-axis, out-of-focus intensity from these ringing side lobes. But that's one avenue to decouple the thickness of the central node with the propagation length of the beam and still maintain high-resolution imaging over a wide field of view. Alternatively, we explored structured light sheets based on different types of square or hexagonal lattices that now provide an entire plane of illumination, but still allow us to decouple the propagation length of the center node from the overall axial resolution of the field of view. And this is what we had termed lattice light sheet microscopy. I'll go over this fairly quickly because both of these have been published now. A recent analysis from a postdoc, Yushi and my group, published last year, and then another paper that came out from Eric Betzig's group and Gokul at University of Berkeley in Science Advances that looks into this exact trade-off between what do we gain and what do we lose when we start to use structured light sheets for light sheet imaging. And just to summarize one example that we looked at here, these are light sheets that are about 20 microns long in total that would be optimized for high-resolution imaging of single adherent cells. This is a computational simulation shown here, although I'll show some experimentally measured data sets in a minute, of a Gaussian beam. And what we can do is we can compare that to one of our commonly used lattice light sheets, which is a multi-Bessel square lattice with these particular NA combinations. With that, we can look at a line cut through the propagation axis, and we can see that they drop to their half intensity at about the exact same point. So these are truly comparable field of views that can be illuminated with both light sheets. And if you look at a cross-section now through the Z direction, you can see that right on focus, the lattice and the Gaussian beams are actually quite comparable. So lattice is in green, Gaussian is in red. But once you get off axis a little bit through this white line here, through the edge of the light sheet, you can see the Gaussian beam is basically diverged to about a factor of 2. So it's twice as wide and has lost about double the resolution that you would get axially. Lattice light sheet actually displays a different type of diffraction profile. So the center node here is actually still quite contained, but there's an increase in out of focus background. And so we wanted to look a little bit more at what this trade-off implies. First thing we did is just look at real space simulations of what your point spread function would look like in these two cases. And again, right on focus, they're very, very similar. But when you move off focus, you gain about a factor of 50% in the lattice light sheet over the Gaussian because it doesn't diffract, or I say diffract differently than the Gaussian beam. This can be viewed a little bit more precisely in frequency space. So real space images are very intuitive to grasp, but they don't necessarily capture the amount of information that's transmitted by the system. So if instead of looking at the Fourier transform, we look at the optical transforms, you can see right on focus, they're quite similar. This is through the red box here, looking at the OTF. But the lattice light sheet has these additional flanking side lobes that provide access to higher spatial resolution. And this difference becomes even more pronounced if instead of just looking right on axis, you look a little bit towards the edge of your field of view, where you can see we still maintain the full frequency support in the lattice light sheet, whereas in the Gaussian system, it's lost. And I think you can see this probably, hopefully with the contrast here with the lights on, in this real space image of looking through a cross-section through the nucleus of the Gaussian beam and the lattice light sheet, where you can start to resolve these axially separated chromatin bundles. Now, the trade-off that you can probably also see in the real space image is that there's an additional background here in the lattice light sheet that's not present in the Gaussian beam. And if we look at the cumulative energy, if we cross basically the cumulative energy propagating from the light sheet out, we can see that the Gaussian decays very quickly. About 50%, the lattice light sheet's quite comparable, but then there's a slower out of focus background decay. And this is one of the trade-offs that we see in lattice light sheet imaging. This might imply some things about photo bleaching. It's actually a little bit more complicated in terms of if you can recover some information from these out of focus profiles versus not. But if we assume that you cannot recover any information from the out of focus side lobes, then we see that the Gaussian beams have about a 10% better photo bleaching rate than lattice light sheets at a cost of about 30% reduced resolution at the edge of your field of view. So this was published a few years ago. I'll just summarize this part very quickly and say our general findings looking both at simulations and numerous experimental systems show that Gaussian light sheets give you a lower background at the beam focus because you've confined all of your energy just to that narrow central node. And in some cases, they can give you about 10% lower photo bleaching, whereas structured light sheets based on different forms of optical lattices in our hands give us better beam uniformity, larger field of view, and higher axial resolution over that entire field of view. So that's kind of the technical part. We've continued developing. This was one of our original prototypes back in 2014. I'm not going to talk in depth about this here, but we've worked on another form of lattice light sheet microscopy called the mosaic microscope, multimodal optical scope with adaptive imaging correction. It's a lattice light sheet microscope that covers a much larger field of view. It works with conventional 25mm cover slips. It has centimeter size scanning area. We realized that we have all of the optics required for this instrument. We essentially have all of the optics required for a number of different microscopes, so we can just reconfigure how we send the light through the instrument. And so that was sort of a rendering of the CAD diagram, but it's a single instrument that can perform, for example, lattice light sheet, expansion lattice light sheet, 2-photon Gaussian or Bessel light sheet, 3D single molecule localization, 3D line scan confocal. I won't go through the whole list. This is one example of just showing a large field of view 3D lattice light sheet microscopy image over about 24 hours of cells expressing a marker for endoplasmic reticulum and histone H2B. So every one of these every one of these time points is in fact a three-dimensional volume. And rather than go into the details and specs of resolution, I want to highlight one of the problems with this, and that's by eye we can generate pretty movies, and we can potentially observe interesting events, but it gets very challenging to actually extract quantitative information from this vast amount of data. So this is about 45 terabytes from a single movie. And in fact, most of the time, we're not interested in all of the cells all of the time, but potentially rare cells in this population that are doing something useful only part of the time. And this is something that Lucas hinted at in his talk last night about event-driven or smart microscopes. So we wanted to take this approach with the mosaic instrument in our lab. And one of the challenges with most of these light sheet microscopes is that, as you can see, the same advantage where you can obliquely section through a small region of your cell is actually a limitation as well, because when you're browsing through your specimen of interest, this is actually what you see. So you see one oblique section through some small slice in your specimen. And it's very difficult to get a large overview of the entire field of view. And if it is even capable of getting a large overview of the entire field of view, it can be difficult to hone in on an exact cell or exact field of view that you want to image. So one of the things we started exploring independently in my own lab recently was, can we use some of the same technology that people use in self-driving cars to do on-the-fly image analysis and extract information from large populations very rapidly, and then use these to home in on cells of interest that we may want to image more efficiently. So there's a bioarchive on this. It should be published shortly, so I'll go over this quickly and maybe just jump to some of the conclusions to give a little more time on the very last project. But this is what it looks like in real time. So this is the Mosaic instrument imaging through the bottom inverted epifluorescent microscope. And this is scanning through the stage at about 20 millisecond exposure, scanning through roughly a square centimeter of volume on the cover slip. And what I want to highlight is that as it's scanning in real time, it's localizing and classifying the XYZ coordinates, in this case an example targeting different phases of the cell cycle of every nucleus in this volume. So an advantage with this is in about 10 minutes you can load the cover slip on the microscope and then get population level statistics of what type of cells or sample are on this cover slip. So this is an example. This is actually from 3 different cover slips averaged together, but we image about, what is that, 18 divided by 3? So 7. 5, 6.5, 6, 60,000 cells on a given cover slip. You can see that the vast majority, 99.9% of them, are all in interphase with this one particular application. About 0.3% are in prophase, which means it's about 1 out of every 5,000 cells. And if you're trying to image something, a specific effect of cells on prophase, this can be extremely challenging to actually quantify and extract. So in about 10 minutes, we get population statistics over very large fields of view of several 100,000 cells. You can do this not just for cell division, but for things like looking at immune synapse formation between T cells and target cells. This is an example where we varied the antigen presentation strength in the target cells, and then we quantified the number of T cells that were either not contacting but not forming a synapse, or contacting and forming a synapse. So you can think of this kind of like flow cytometry level of information where you can very, very rapidly scan through large populations of cells. But rather than just quantifying information about fluorescence intensity, you can actually quantify information about image characteristics that tell you something unique about what those cells are actually doing. But an advantage of this is that not only do we have the population level information, we have all of the coordinates of every cell that we wanted to image. And so you can go back then and sample in high resolution and multicolor 3-dimensional volumes with subcellular resolution of the rare cells that you wanted to look at. You can also use this not just in fixed samples, but in live samples to say, I want to find that 1 out of every 5,000 prophase cells, and then capture a dynamic movie, in this case, looking at kinetochore localization and distribution throughout an hour-long time course of mitosis. So in this case, it's not quite as high throughput because we focus on one cell for an hour to capture the entire mitotic process, but you can load your cover slip in the evening, come back in the morning, and have a dozen movies of cell mitosis quantified all the way through cell division. And that can be useful for looking at, for example, screening the effects of different drugs on kinetochore velocity. This is just an example of one that we knew would have a clear effect of Taxol. And finally, I wanted to highlight that this is, say, cell division is one of the prototypical examples that people target for smart microscopy because it's characteristic, it's involved in every proliferating cell type. But we can adapt this in a matter of this was about 3 days to adapt the network to a new application looking at immune cell, cancer cell synapse formation in response to different antigen presentation strengths. And we think that this is a way to rapidly acquire high-resolution replicates or movies of very rare samples that have typically been challenging to image because you'd have to sit on the microscope and it would take you an hour and a half to find the one thing you wanted to look at. It could also be extremely useful for things like expansion microscopy, where now your volume is much, much larger, but you're still only interested in this one rare event over this very large volume. Okay. So a little bit of a whirlwind through instrument development, applications. I'm going to focus the rest of the talk, and I don't have a clock, so I'm just going to guess that I'm on time until someone tells me I'm not, on our single molecule imaging experiment recently. So a few years ago when I started the group, we got interested in part from collaborations with Javier Darzak's group and Bob Tejan's group looking at transcription factors navigating the nucleus using our light sheet microscopes. And we became very interested in, can we use our instruments to tackle some questions that haven't been tackled before in this field? And to give a little bit of very brief background, this is a dramatic oversimplification of lots of biochemical analysis. But if we started looking at a nucleus either in electron microscopy or in epifluorescence microscopy, we can roughly categorize chromatin in the nucleus. So there's 2 meters of DNA that are coiled like a ball of yarn into about a 10 micron diameter sphere in the nucleus of every cell in our body. That compaction results in a heterogeneous distribution of chromatin, and roughly we can classify this as euchromatin, which biochemical assays have shown us is open, accessible, loose, it's active, so expressed genes tend to be there, and it's about 80% of chromatin. And this is sort of the light staining in electron micrographs, or the dark staining in DAPI staining. And we can compare this to heterochromatin, which is dense, closed, inaccessible, it's compact, it's inactive, and it's repressed. So this would be like centromeric repeats or telomeres in chromatin, as well as repressed genes that are not on, say, in differentiated cells. And these are dense regions in electron micrographs and bright regions in DAPI staining. And so coming from an engineering background, with the instruments we had, we started with a very simple question, and it's, could chromatin specification, the biological function of these two different compartments, arise perhaps from some change in the biophysical environment in heterochromatin versus euchromatin in the nucleus? And say, can biological function in fact arise from changes in the material properties of these two different regions in the nucleus? And so to approach this, I'm going to highlight some work from, again, from Yu Shi, who's a postdoc in my group, and from Tim Doggard, who was actually my very first graduate student in the group when I started. And we approach this using single molecule imaging. So this is an example of single molecule imaging on the lattice light sheet microscope, looking at slowly scanning in 3 dimensions through a Z-plane, and this is tracking individual nucleosomes with halo tag H2B in the nucleus of cells. So this is standard sort of bread and butter single molecule tracking. What's interesting is on the same instrument now, we can perform high resolution 3-dimensional lattice light sheet microscopy. This is actually the exact same cell, except that in this case it's labeled with a photoactivatable PA-JF646. And in this case, it's labeled with a non-photoactivatable PA-JF525. And this gives us information now about the single biomolecule fluctuations of nucleosomes and the underlying chromatin density that those nucleosomes are moving within in the local area of the nucleus themselves. So we can take, now this is a single plane through our 3D volume. We can put this through an image classification protocol. Happy to go over the details of this maybe in Q&A or after the session. We can partition chromatin into, say, 7 different density classes. 7 is actually a nice partitioning here because it maximally explains the variance without overfitting. And for sake of the next few slides, very dense heterochromatin regions, and I'm using heterochromatin admittedly very loosely because we're simply quantifying chromatin density, not, say, post-translational modifications one would normally associate with heterochromatin. But chromatin dense regions are going to be classes 7, 6, and 5, and then chromatin very sparse regions are going to be 1, 2, and 3. And so immediately we can see that the majority of the nucleus is actually occupied by low to intermediate density chromatin, and very, very low density and very, very high density are occupying proportionally less of the nucleus. When we combine this with our single molecule imaging approaches, what we can see is we can localize individual nucleosomes with a spatial resolution of about 20 nanometers laterally and about 80 nanometers axially. And we can overlay this with the given chromatin class that the nucleosomes are diffusing within. So this gives us a mechanism then to explore the question of whether the biophysical fluctuations of nucleosomes are different when they're in dense chromatin classes or sparse chromatin classes. So how does the local density of chromatin in the nucleus affect the biophysical fluctuations of nucleosomes? To give a little more detail on how we extract information from that, what we do is we plot the mean squared displacement of individual nucleosomes as a function of different time lags. We're imaging at 20 millisecond frame rates, and we image typically a single track on the order of about half a second. So we're probing very fast timescale fluctuations of nucleosomes in these environments. From this, we can then plot this on a log-log plot and extract out the slope of the plot, which gives us a power law exponent. In this case, it's, apologize for the nomenclature here, it's alpha. And we can extract basically an apparent diffusion coefficient of how much the molecules and how fast they're moving in that given environment. So I'm going to show plots of the apparent diffusion coefficient and the MSD exponent in the next few slides. And to give a little bit more intuitive sense of what the MSD exponent implies, it's a bit of a measure of anomalous diffusion. So it's say, how does the molecule diffusion compare to what one would expect for free Brownian motion in a given environment? An alpha value of less than 1 indicates some type of confinement or trapping. An alpha value of equal to 1 looks like something like normal Brownian motion, and an alpha value of greater than 1 would indicate some kind of directional persistence or directed motion. So if we plot the apparent diffusion coefficient and the alpha exponent for nucleosomes in different chromatin density classes, we can see right away that there's a clear trend where nucleosomes in these denser chromatin regions are, we'll say, they're not necessarily more confined, so they don't display more anomalous diffusion. It's all confined diffusion, but they move slower and they move less. So they appear to move less, they're less mobile, they're less diffusive than nucleosomes in these low density chromatin regions. So this was an interesting observation because it says there is something fundamentally different about how nucleosomes experience their local chromatin environment in these two different compartments. But we wanted to ask, how can we explain this? And so we can come up with maybe three potential explanations. So there's variations in the viscoelastic properties of the interchromatin space. So maybe they're moving within a more viscous environment, and that's why they move slower. You could envision variations in nucleosome packing. So maybe they're spatially packed or organized slightly differently, and that allows them to move differently. Or we could look at variations in active processes that tend to be spatially localized in these different chromatin compartments. So we wanted to explore these one by one. So to look at variations in the viscoelastic properties of the environment around these different chromatin regions, we looked not only at individual nucleosomes that were tethered now to the chromatin chain, but free diffusion of halo tag proteins that are navigating through this interchromatin porous meshwork. And that tells us something about the viscoelastic properties of the chromatin space in these active, we'll say in these dense versus less dense compartments. So interestingly, when we look at free diffusing halo tag and we plot in this case, apologies, this is the roughly equivalent to the apparent diffusion coefficient. It's an old plot I didn't update yet. But this is the mean squared displacement at a time lag of 0.1 seconds. We can see there's actually almost no dependence on chromatin density class in the nucleus. So a single free diffusing molecule at the length scale of about 2 nanometers does not experience any different diffusive behavior, whether it's in a dense chromatin environment or a sparse chromatin environment. It essentially sees the whole nucleus equally. So it doesn't look like there's something different about the viscoelastic properties of the interchromatin space in these different compartments. And so that brought us to our second question, which is there any difference in chromatin packing? And so to do that, we switched microscopy modes. This is actually a microscope we built in the lab that's based on a swept high-low instrument. This is using some of Luke Lavis's newly developed spontaneously blinking Gennelia fluor dyes. This is an example of JF630B. I think he talked about that briefly during his seminar. I will say these things are amazing. So this is a halo tag coupled ligand. You label cells with it, you fix it, you put it on the microscope in PBS, and this is the type of images that you can get. Pop them in the fridge for a week, pull them back out of the microscope, or pull them back out of the fridge, put them back on the microscope. It looks exactly like this. There's no redox buffer, there's no converting to a dark state. It's just put them on the microscope and twinkle, twinkle, little star. So this is an example now using this to do 3D localization microscopy of halo tag H2B. We can do this, we can put it through our standard localization microscopy pipeline, localize them in 3 dimensions. What we do here, because we want to compare this to chromatin density classes we had seen in our live cell imaging, is we can convolve with the lattice light sheet point spread function, deconvolve with the conventional lattice light sheet point spread function, and then put it through our normal classification network. So we recover basically chromatin density classification at the same length scale that we have in our live cell imaging. And then we can classify each individual molecule based on the local chromatin density around it. And I'm going to go into a little bit of detail on how we quantify differences. So we put this through what's called a pair correlation function or Ripley statistic. And to give a real quick background, if you have completely randomly distributed spots, this curve would give you an equivalent value of 1 at all length scales measured. If you have a deviation from random, so an increase, for example, in your pair correlation statistic, it indicates clustering of molecules at a given length scale. So we can examine if there's differences in clustering. And I want to highlight this is actually density irrespective. So this says, yes, we know they're more dense because it's in a higher intensity, more crowded region. But this looks at basically the arrangement of spatial packing and its deviation from a random organization, irrespective of density. So what's interesting is if we look at these pair correlation statistics for H2B molecules in different density chromatin domains, we can see that there's a steeper slope and a larger deviation for sparse chromatin classes. And our densest chromatin classes look closer and closer to the random distribution. And all of them tend to deviate at a length scale of around 200 nanometers, which suggests functional specification or deviation in random architecture at the length scale of around 200 nanometers. Now, one of the things that's interesting from this is this linear slope of this pair correlation statistic, when you plot it on a log-log scale, implies something about the spatial organization of the points we're observing. And in fact, it implies a fractal architecture, which just means that it displays self-similar architecture at various different length scales. So one of the things we can do with this is we can fit the slope. Okay, so yeah, so the power law here indicates fractal structure. A fractal structure of 3, so I'm jumping over a lot of things here, but fractal structure of 3 indicates optimally packed uniform distribution. So it'd be like you had a bunch of marbles in a jar that are optimally space-filling and randomly packed. A fractal dimensional of 1 would be like all of those marbles are in one line existing within your jar. So they're highly non-optimally space-filling. And so what we can do is we can fit basically the slope of this plot on a log-log scale, extract the fractal dimension from the dimension minus alpha, or minus a, which is the power law exponent, which is the slope of this line, and it tells us something about the spatial organization or spatial packing of nucleosomes in these different density chromatin regions. So if we look kind of on average, I'll just pull one example here. This is not unique to us. This is using a localization microscopy. We see a fractal dimension of about 2.2. This is actually in large agreement with the fractal dimension of chromatin measured with a variety of other imaging technologies. What's interesting, though, is if we plot this as a function of chromatin density class, and we see a very clear trend that nucleosomes display different physical packing in these different chromatin density environments, regardless of their underlying density. So they do seem to utilize different packing strategies and different organizational principles in sparse euchromatic versus dense heterochromatic regions in the nucleus. And what we find is that nucleosomes in sparser environments are less randomly packed. They're more structured than those in dense environments, and those in dense heterochromatin environments are more randomly packed and optimally space filling. How much time do I have left? It's like, don't say -10. $10. Fantastic. Okay, great, cool. I'll go a little bit slower. Okay, so we did see that there are variations in nucleosome packing. This doesn't necessarily tell us that these variations in nucleosome packing can explain this change in apparent diffusion coefficient that we see. We just said there are differences here. And so we wanted to ask, is this a potential explanatory phenomena to predict what the nucleosome apparent diffusion coefficient would be? And to this, we turn to adopting existing biophysical models. So, bottom-up modeling of chromatin from a polymer standpoint is a very well-developed field that is outside of my exact area of expertise. So we partnered with John Crocker at University of Pennsylvania and others who are polymer physics experts. We adopted a previously published model from Shinkai et al. And Togashi, published in PLOS Genomics, PLOS Computational Medicine a few years ago. And one of the things that I like about this model is that it assumes a fractally organized Rouse polymer diffusing within a viscoelastic medium. So this is a very basic bottom-up polymer simulation approach. I'll go over I won't go over the exact derivation from it, but one of the things we like about it is it provides an analytical formula to predict the nucleosome mean squared displacement and the apparent diffusion coefficient and the alpha exponent based simply on biophysical measurements that we can make. So this is the full derivation. I'm going to say don't worry about the full derivation here. These are the two main terms that we're interested in. So this is the mean squared displacement of our nucleosomes. This is the apparent anomalous exponent of the nucleosomes. And essentially, all of the derivation here from the bottom-up polymer model relies on measurements of, say, the scaling of free protein diffusion within the interchromatin space, which is a measurement that we have experimental measurements for. The fractal dimension of nucleosome packing in the cell, which is a thing that we have experimental measurements for. The estimated local nucleosome density in the cell, which if we measure the expression level of our halo tag protein in the cell, we can get now from our localization microscopy data sets, and the radial size of a chromatin domain that we can extract now from our pair correlation curves. So the unique thing about this that I think is quite interesting is that this is a bottom-up polymer model that has absolutely no free parameters to fit to our experimental data. Every parameter in the model is defined by experimental measurements in our system. And if we put this through our pipeline, we can use this now to predict what the apparent diffusion coefficient would be for nucleosomes in these different density chromatin environments. This is our experimental measurements in control cells, and this is what our polymer model would predict. So, not having done a lot of modeling myself, but done a little bit of modeling, I find it actually quite striking that it even matches remotely agreeable with no free tuning parameters. But we can see that, and I'll say this model is based on purely thermal fluctuations in this polymer model. So it has no active processes, no consideration of, say, nucleosome attractive or repulsive bonds. It's simply thermal diffusion of a Rouse-like polymer. It actually does a quite good job matching in these dense chromatin classes, but it predicts a larger than expected scaling as a function of chromatin density. And specifically, the model would say we would expect nucleosomes to be a lot more mobile in these dense chromatin compartments than we actually observe experimentally, which was a little bit confounding. So we wanted to go back and look at this in a little more detail. And so we said, could this deviation from our model, which only assumes thermal processes, be explained by active processes that are compartmentalized into different regions of the nucleus as a function of chromatin density? And this is actually a very interesting area of research where I think there's a fair amount of, I won't say controversy, but I'd say conflicting studies in the field. So there are several papers that suggest that in these active transcriptional compartments, there's an active stirring or thermal motion due to RNA polymerase and all of these other active processes that could locally stir chromatin and increase its mobility and basically make it breathe a little bit more, make it more accessible to things like transcription factors and polymerase. In contrast, another model would say, well, a lot of these transcriptionally active molecules have intrinsically disordered domains, and they can form condensates or hubs that locally alter the material properties and attract chromatin and basically stabilize them. And so we wanted to look at this. One of the, I'll back up a little bit and say that the model for locally stabilized chromatin is not unique to things that we've been looking at. It's been suggested, actually, when you inhibit transcription using pharmacological methods and look at nucleosome motion on the whole cell level, that if you inhibit transcription globally, nucleosomes everywhere in the cell get more mobile. So this was suggested to say, actually, transcription may form these hubs that locally tether chromatin into this stable compartment. But this was measured on a whole cell level. So we wanted to ask, is there any local specification to this tethering effect that might be present in our data? And so we took a range of pharmacological perturbations and essentially applied the same assay to say, how do these different perturbations affect different chromatin density classes, and how may they uniquely alter chromatin motion or nucleosome motion in these classes? I'll just show two examples of alpha-amanitin and TSA shown here. So if you inhibit transcription with alpha-amanitin, you preferentially increase nucleosome motion in low to intermediate density chromatin regions. So blue is our control curve here, and orange is our alpha-amanitin transcription inhibited curve. So almost no impact in the apparent diffusion coefficient of nucleosomes in these high density classes, but a dramatic increase in nucleosome mobility in these low to intermediate classes. And this would suggest that transcription predominantly occurs in these low to intermediate density classes. In contrast, if you hyperacetylate histones with triclostatin A by inhibiting histone deacetylases, you decompact dense chromatin, but you have very little impact on sparse chromatin. So this says, yes, there's unique biological functions that are focused in these different chromatin density classes. We can go in and actually look at well, this suggests that transcription is happening in low to intermediate density classes, but we can directly measure that with, for example, an MS2 stem loop system to look at transcriptional bursting. These are cells we got from Joe Rodriguez at NIEHS to look at estrogen responsive gene TFF1. And we see that almost all of the bursts for this gene occur in these low to intermediate density chromatin classes, which suggests that this is really where transcription is happening. Okay, so yes, there are variations in active processes that are localized in different chromatin density regions, and we wanted to see what does this do in relation to our model. So this is an example where this is our normal control cells. This is what the model predicts based on purely thermal forces, and this is our transcription inhibited experimental data, and this is what the model would predict based on purely thermal forces. So if we inhibit transcription, we inhibit the active process of transcription, actually the entire nucleus seems to behave like a thermally driven Rouse polymer, but that we don't. But if we actually want to model this from a bottom-up standpoint, we need to take into account a stabilization effect of nucleosomes in these active transcriptional compartments. So I think I'll skip over some of the overall summary of this. In sparse regions, we see faster nucleosome motion, more clustered nucleosomes, and dynamics that can be explained by thermal and active forces. And in these dense chromatin regions, we see slower nucleosome motion, more randomly packed nucleosomes, and dynamics that can be largely explained by thermally driven polymer fluctuations. Nucleosome and spatial organization vary as a function of chromatin density in the nucleus. Polymer models predict greater than expected dependence of nucleosome motion on the local chromatin density, and transcription predominantly occurs in these sparse regions. And our data support a model where these transcription condensates may locally stabilize chromatin into these active compartments over time. What's interesting is the viscoelastic properties of the interchromatin space appear to be completely independent. So it's not like they're just making a stickier, more viscous thing and that's sort of tethering everything together. From a free diffusing molecule, it seems to be completely agnostic to the chromatin density. And that would just say, well, you know, the nucleus is full of a lot of other things besides chromatin and RNA and other nucleic acids and other proteins. So the viscoelastic properties appear relatively constant. But this is one way that provides a mechanism to stabilize genomic regions in these active compartments through a nucleosome-specific stabilizing effect, potentially through things like BRD4, bromodomain proteins stabilizing acetylated histones. But it doesn't actually seem to reflect any fundamental change to the viscoelastic properties in these different chromatin environments. So I was hoping this would be up on bioRxiv before I talked today. It's uploaded, but not yet approved. So it should be actually uploaded next week, and you can have the full story there. I'm also happy to talk about a little more questions that you might have and get a little bit deeper into the story. With that, I'll wrap up and summarize. I talked a little bit about some of the microscopy work we're doing, about some of the AI machine learning approaches to automate imaging, and then focused a little bit more on our latest story on using single molecule imaging to explore chromatin architecture in cells. With that, I want to highlight really all of the people that did the work. The AI machine learning project was a joint project between Yuxi and Jimmy Tabet, who's an MD PhD student at UNC Chapel Hill. And the nucleosome imaging project was a joint project between Yuxi and Tim Doggard. Yuxi is on the faculty job search market now. He's fantastic. Tim Doggart is on the postdoc search now. He's also fantastic. I'd be happy to tell you more about both of them. Thank you to all of our funding sources that allow us to work on a variety of different projects. We're always recruiting, so anyone interested in using the instruments, building new instruments, I'm happy to talk to anyone interested in postdoc or grad school positions. And with that, I will finish, and hopefully I'm still on time.
Beautiful talk. Just a little question for your smart microscopies. The software you develop, are they really dedicated to the instrument, or is it something larger that later you intend to distribute to community, or what's
that's a great question. So if I can rephrase the question, it was, is this unique to Lattice Light Sheet Microscope? Does it work on other instruments? And I'll say it's a bit of a trade-off. So the image analysis one, it's just a Python script, and it's using YOLO version 5, which is a commercially available package that you can download and train. And so we actually purposely decoupled that from the Lattice Light Sheet control module. So it's integrated in the sense that Lattice Light Sheet Microscopy control module will call the Python script and run it, but it's not like completely integrated in LabVIEW specific fashion. So anyone can basically download that code, set it up on their own instrument, but they'd have to do the integration themselves. Anyone that wants to run a Lattice Light Sheet Microscope can basically run the same software that we have now.
Hello, thank you. I had a question actually regarding your different subclasses, so 8 subclasses, and I was wondering how it correlates with other studies that were published. Actually, we had a talk from Christine Bistricki, I don't know if she's still here, and she, with Heidi method, I guess you know that, she finds A type and B type chromatin, So here, did you just, how did you actually define your 8 classes, and does it correlate to some biological functions or?
Yes, so that is a fantastic question. I'll tell you the true story of how we picked it, and then I'll tell you the validation that said it was actually an appropriate metric to pick 8. So the true story is there were other papers that had published this. So there's a paper using structural illumination microscopy that used a similar approach and looked at immunofluorescent distributions of active and repressive proteins. They picked 7. So we started with 7. I will say this is a completely artificial partitioning of a continuous distribution in chromatin density. So I don't want to say that the boundaries between these different density domains imply anything physical about boundaries between different densities in the cell. It's completely artificial. 7 actually works out, and I have the supplementary slide in here, but I won't jump to it for the sake of time, that if you look at like Bayesian information criterion or Akaki information criteria, it says 7 is about the optimal fitting in distributing this Gaussian continuous distribution to explain the variance that we see without overfitting the data. So that doesn't mean that like if we split from 2 classes or 3 classes or 14 classes, it would fundamentally change the analysis. It just says from an information statistics standpoint, this is an optimal value to pick. I will say we did look at partitioning into 3 classes or 14 classes, and the trends are basically the same, except that the more and more classes you split into, the noisier the data gets because the fewer molecules you have in a given class.
So, but for example, can you make a correlation between your 8 classes and which one is the A-type compartment and the B-type compartment? Because here it seems that you don't correlate to what was done before.
You're absolutely right. And I want to hesitate to say any correlation between A compartment and B compartment from the data that I've shown here, because those are traditionally defined based on ChIP-seq data and RNA-seq data to say what's basically like the principal components based on what one would see from transcription factor binding, RNA polymerase binding. And so A and B compartments are not necessarily classified, well, and from high-C data, of course, not from, say, images of density, but from high-C data. So we have no genetic specificity here. I will say that if we come in and we have another project doing super resolution DNA paint, then we can actually see partitioning in A and B compartments at different densities, but we haven't looked at exactly how that distributes in the 7 classes we've looked at here.
We still have time for more questions.
Thanks for the really nice talk, Wes. This may be a naive question, but your map of spatial density, the chromatin density, is relatively low resolution, and the single molecules that you're tracking, I mean, you're making the assumption you see it in this region, but I'm wondering if there's any possibility of bias for your halo tagging, that it could be biased towards tagging the low resolution, the low density regions more than the high density regions, and whether, yeah, if you thought about that.
Yes. So the short answer is yes. So one of the things we had done is we just do our halo tag labeling together with hooks and look at the overlap and correlation between the two signals, and we see essentially that it goes everywhere with no bias. That's the short answer, but the better part to your, not to say the better part, the other part to your question is what the heck do we know about like an individual nucleosome, which has a length scale of about 8 nanometers, diffusing in this density environment that we measure at a few hundred nanometers. So the level we're measuring density is an order of magnitude larger than the nucleosomes themselves, and 2 orders of magnitude larger than the individual biophysical fluctuations. So it's like you're flying over a mountain range and you see topology variations, and like you have a little pebble on the mountain range, and it's like, what the hell does it know if it's at the top or the bottom? It just sees the ant next to it, right? So that's an actually open question. I don't want to say that we're uniquely measuring. I want to say the measurements seem to suggest something functionally partitioned at a length scale of a few hundred nanometers. So there is a difference that we can see in a chemical environment at the length scale that we can measure, but it's unclear how to exactly tie the nucleosome fluctuation to what that density is.
Very quick question: Did you observe transitions in the diffusion of nucleosomes going from one of the classes to the next?
Yeah, that's a fantastic question. So in this system, no, because our tracks are too short. These are using PA-JF646 dyes. Actually, they're fantastically photostable, but in this imaging condition, because of how we were imaging, we were basically like, we were imaging 50 frames, and then we were stepping to a different Z slice, and then stepping to a different Z slice, and stepping through. So we weren't focused on imaging really, really long tracks. On the swept high-low system, we can actually image not using PA-JF646, but just using regular JF646 for like a minute of a single molecule track. And these are histones, so they don't move very much. But there we actually do see transitions between states in a single trajectory, and that's something we're looking at in more detail now.
Do we have other questions? I actually have one question about the smart microscopy to detect events. How sensitive is that to acquisition conditions, to exposure time, to excitation, irradiance, and these kind of things?
Yeah, so that's a great question. It's reasonably sensitive. I'll put it that way. I mean, so obviously if you give it a sample it's never seen before, it will perform terribly. And I jumped over the conclusions because most of it's quantified in the bioRxiv article and should be out shortly. So under ideal conditions, we're like 80% to 85% efficient at classifying things in the appropriate class. It's sensitive to things like defocus. So as we scan around, we actually mapped the topology of the cover slip and made sure we are always in focus. We looked at a little bit of signal to noise, but not that much. So it's reasonably robust to signal to noise, partly because it's been trained on cells that have varying expression levels, and so it sees that in its normal analysis. But I will say if you give it something different than it's been trained on, like different noise levels, different morphologies, then it doesn't perform very well. And actually, you need to be a little bit careful. So one of the problems with the Taxol effect was it changed the morphology of chromosomes in metaphase so substantially that cells were basically locked in metaphase, but the algorithm was not predicting any cells in metaphase because they looked very different than control cell metaphase. So the algorithm just tells you what it's been trained on. It doesn't tell you what the actual true biology is.